To begin with, a signal transduction occurs in liver cells and that contains a hormone called a ligand. A ligand is a small molecule that is attached to the metabotropic. The ligand needs to attach to this receptor to enter inside the cell. Once the metabotropic binds to the ligand, it goes through a process called protein conformation. Protein conformation means to change the shape of the protein. Right beneath the metabotropic is where the heterotrimeric G protein is located. The G protein contains 3 subunits, which are alpha, beta, and gamma. At the alpha subunit, GDP is attached to it. The alpha subunit then switches GDP to GTP. Later on, the alpha subunit binds adenyl cyclase, which is an inactive enzyme during this time. When alpha gets attached to the adenyl cyclase, it becomes active and then is ready to convert GTP to CAMP. In this process, 2 phosphate groups will be taken away. The molecule that speeds up this process is called ATP. ATP is then attached to the CAMP is called a secondary messenger. Later on, CAMP will spread throughout the cell and will find its way to the protein called protein kinase A, which has 2 subunits called catalytic and regulatory. The CAMP will bind to the regulatory and will activate the catalytic protein and will be detached from the regulatory protein. Once it detaches, ATP will bind to it. The catalytic will bind to the phosphodiesterase and will activate the phosphodiesterase. Later on, the phosphodiesterase will break down the glucose, so glucose can be released outside the cell. This is how molecules here communicate with the action called cellular response, trying to get the glucose out of the cell.